Afternoon, everybody. Really happy to be here. So I'm going to talk about some of the work in moving an idea from literally from the bench to the clinic at home in Ottawa for me. I've been a prof for about 10 years. The concept you'll see was first drawn on a board about 7 years ago, and today we're 2/3 finished our phase 1 study. So I'm excited to show you the journey. So we launched Yellowbird with two former grad students who worked on this during their PhDs in 2023 because we were really convinced we had something that could be of diagnostic utility. And the only way to move it forward was to commercialize. So we did that, and we are in Ottawa still. We work out of the Heart Institute. We're at the phase 1 clinical level. And we really focus on solving diagnostic needs. Our first offering is radiopharmaceutical, but we have a pipeline with some MRI and optical agents to come. Nick and Alexia were my grad students who worked on this during their grad times, and they decided that they believed enough in it to jump into forming a company. So they're CEO and COO, and we're joined by Michael Gottlieb, who some of you may know from Point Biopharma, who's also saw some value in what we're doing and has helped us with the business side. I wanted to start with the first case that we've imaged. So this is an FDG scan. This is glucose mapping of a 70-year-old male with recurrent primary T-cell CNS lymphoma. It's rare, but it happens. And so they had a resection. You can see the original site of resection at that blue arrow at the top of the skull. But they still had some kind of neurological symptoms, returned to the clinic, and this is where they started a new investigation. So the FDG scan was systemically unremarkable, nothing to see, even around the initial site, a little halo of FDG uptake, but nothing specific. And so this kind of illustrates the problem we're going after. It's very difficult to see inflammation in the brain and in the heart with current PET imaging options. So MRI is used a lot in the diagnostic workflow, but it doesn't really see active disease. It kind of sees the scars left over. And we want to be earlier, and we want to see this during the active inflammatory phases. So as you saw with FDG PET, it is limited in the brain because the brain uses so much glucose when it's healthy. And in addition, there's a dietary prep that's required. And this is tough for a larger proportion of patients than I knew. So this requires ketogenic diet and overnight fasting. And if this is not done correctly, especially in the heart, it's very difficult to interpret the scans. So we set out to look at an alternative. And our alternative was, we call it Nucaviz. It's a fructose-based tracer. So instead of looking at how we use glucose, we look at how we use fructose. And by doing this, we're really hoping to be more specific in how we can map inflammation, especially in the heart and in the brain. So why fructose? So we all know glucose and glycolysis powers the cells, especially in the brain and the heart, all the time. There's a negative feedback loop, that's that red arrow. So if we make too much energy, it shuts itself down. When there's a disease process going on, the cell has figured out that if it switches to fructose, it can overcome that feedback inhibition, and it can actually power more of its processes that it thinks that need to happen. So the nice thing about fructose, as opposed to glucose, is fructose is really only metabolized outside of the liver when things are going wrong, right? It's a really big powerhouse for inflammation, but not much for regular daily function. So we decided to radiolabel fructose, and we did this using the enzyme kinetics of one of the key steps in the pathway. We radiolabeled at the 4 position, which gave us the ability to trap the product in the cell and basically just mimic how FDG works. It gets transported, it gets phosphorylated, and then it gets stalled and trapped and accumulates. So again, back to this is the FDG scan of our first patient. So this was done in compassionate use study in Germany with Ralph Bundschuh in Dresden. And this is the first Nuclear scan that was ever recorded. We wanted safety and we wanted to see a little bit of decent biodistribution. We never anticipated the outcomes, which was not only did we see the inflammatory region around that initial site of resection, which you can see clearly at the top of the skull, but if you look at the sagittal view, that white arrowhead is pointing to a new cancerous lesion. And so this lesion would have been missed by traditional PET scanning, and the patient would have been sent home. But because we found this, the clinicians put the patient back on methotrexate, and we were told that the lesion is gone. So this was super exciting for us, and it really proves out the case that if we target metabolism associated with disease, we can actually get really nice signal to noise and be able to pick out these very small lesions. So they also did a PET-MR, which we're really happy about. With compassionate use, we have no say on follow-up, we have no say on data, we just hope that it's injected properly and we get the scan. So this was awesome. So it's a T2 weighted MRI on the top, and you can see the white arrowhead points to a little bit of hyperintensity, and this T-cell CNS lymphoma is hyperintense on T2 weighted MR, and you can see it overlays really nicely with the Nuclear uptake on the PET. So it really kind of supports, based on the initial clinical diagnosis, and we have recurrence, that the co-localization is actually tumor related. So this was all really awesome. We've since moved on to do a few more patients in Germany. We have vascular graft infection mapping, and we even think we have some kind of inflammatory disease of the colon that's lit up. And we're currently hoping for a few more brain cancer related images from our partners in Germany. But basically what I hope to have shown you from that first patient scan is that, we thought about the disease pathway, we thought about the cellular energy component, we labeled it in a position that allows for biochemical trapping, and this gave us the ability to see with really high resolution, signal to noise resolution, that these really small lesions and small areas of disease. So this is really kind of a pan-disease platform. We really believe this is any inflammatory condition will light up this with this tracer. If you look in the literature, most diseases actually have a fructose phase built into them that doesn't necessarily initiate the disease, but does exacerbate the pathology. This is a phase 1 asset, and I wanted to show how we went from clinic to where we are today, and then where we're going, just by this map. So we had our preclinical campaign in toxicology with my academic hat on, and then after that, we moved it to Yellowbird after patenting with the university. The University of Ottawa is very startup friendly. They have really awesome licensing and reassignment of the IP back to us, as well as support with lab space. CMIE came on at that point, helped fund the CMC package. We also had to reinstitute GMP at our radiochemistry facility locally in Ottawa, which was a larger undertaking than any of us thought it would be, but it's done and the system is in place. And so with that, we were able to move through CTA approval for the phase 1 and initiate that in Ottawa. We hope to move forward now to phase 2 in a cardiac and neurology mixed bag trial, hopefully, since we think we can attempt to hit both of those disease areas with our radio tracer. We're expecting approval by 2030. I think everybody in the room knows radio tracer development is really cost effective relative to drugs. So with about $50 million, we can move through to final approval. I wanted to also mention, in addition to CMIE, Ontario and Canada, we're really fortunate to have a lot of really friendly startup funders for small companies. So locally in Ottawa, we have Capital Bioventures, we have Obio, we have Facet. So they're all listed in the corner, and all of the funding that has come from these different sources has really helped us to get to where we are today. So as of today, none of the founders have put any personal money into the company, which is awesome because that's not an option for me, my wife told me. So it's good. So I want to just finish up. I want to show you the first of our phase 1 trial. So this was 12 healthy volunteers. We were looking at dosimetry. We want to do full PK. But we also wanted to compare it to FDG. And so this is the first person scanned, the fructose on the left, FDG after the standard of care fasting regime on the right. And what you can see is a relatively clean scan. There's liver metabolism, of course, as a primary site of fructose utilization, but not anything excess of what we see with FDG, primarily clearance through the kidneys and bladder, no brain uptake, and no heart uptake. And so we're really happy with this baseline. I think, I thought I had blood data, it must have disappeared. We've also been drawing blood. We have a arterial input function, and we found that whether it's whole blood or serum, there's no difference, so it's not bound to red blood cells or anything like that. So it looks like a really well-behaved radio tracer, clears in about 4 hours. It's about back to baseline. So a little bit shorter of a scan interval than FDG, but I don't know that that's necessarily a bad thing for clinical workflow. So that's what Yellowbird is up to. Hopefully, next time we meet, I can present some early phase 2 data, but we really hope to advance this as quickly as we can. And again, the support of the provincial and federal communities around radiochem and radiotherapeutics and diagnostics has kind of let us get to where we are today. So I'm grateful for all of that. Thank you.